just discuss. No, we have discussed the neurons, the different structures of neurons, and the functions of neurons. Neurons, they don't exist individually. They are connected with each other. They form neural networks. Eventually, they make up the nervous system. The human nervous system has two branches, the central nervous system and peripheral nervous system. Next, let's discuss the central nervous system. The central nervous system has two branches, the brain and the spinal cord. The brain sits on top of the spinal cord. Let's start from the lower part of the central nervous system, the spinal cord. The spinal cord is a band of nerves. It's a big bundle of nerves, and it's protected by the bony structure of the vertebrae. The vertebrae serves like a casing to protect the spinal cord inside. Of course, the vertebrae also have a supporting function. It supports upper body. The spinal cord is divided into 4 sections. The next section is called the cervical section, the upper back, thoracic section, the lower back, lumbar section, and the tail section, sacral section. Sometimes people get injury in the spinal cord. This may result in serious problems like paralysis. Basically, if a person gets a break in the spinal cord, all the body parts below the break point or below the injury, below the lesion, is going to be paralyzed. And also, you receive, the brain receives no sensation from any point below the break or below the injury. This means the spinal cord is responsible for sending information from the different body parts to the brain. And also it's responsible for carrying information from the brain to different body parts. So in other words, motor neurons carry command control information, instructions from the brain through the spinal cord to different body parts. The torso, the limbs, the fingers, the hands. At the same time, the sensory neurons carry information from different body parts to the brain. If a person suffers a break in the spinal cord, any section of the spinal cord, the parts of the body below the break point will become paralyzed, no control, lost control. And also, the sensory information from any body part below the break point is going to be unable to pass through to the brain. So the person has no sensation or control for the body parts below the break. A case of spinal cord injury is Christopher Reeve, the actor who played Superman. He was riding a horse and he fell off the horse. He broke his neck. So from neck down, he was paralyzed. He was not able to move his arms and legs. His vocal cords was even affected by the break. He was not properly controlling his vocal cord to make speech sounds. And he had no sensation from below the neck. Eventually, he died prematurely, partly related with the injury. spinal cord is very important in communication between the brain and different parts of the body. Besides communication between the brain and different body parts, another function of the spinal cord is reflexes. Spinal cord controls some really important reflexes. An example of reflexes would be this withdrawal reflex. The finger is touching a hot object, like a flame from a candle, and then sensory neurons carry the information. The Greenland sensory neuron carry information to the spinal cord. In the spinal cord, the information gets transmitted into an interneuron. Interneuron is like a connector neuron. And then the information stimulates the motor neuron. The motor neuron gets connected to the muscle fibers. And the motor neuron stimulates the muscle fibers, and there's muscle contraction. The hand withdraws. This process, this loop doesn't go through the brain. You can see that means the reaction, the withdrawal complex, is not controlled by the brain. It's controlled directly in the spinal cord. You usually withdraw your hand before you feel the heat or before you feel the pain. This is a very important protective mechanism of the spinal cord. The pain information, the information about touching something hot, eventually go to the brain, go upward to the brain, but it takes longer than the reflex. We have some other reflex controlled in the spinal cord. For example, the knee jerk reflex. If a doctor taps your knee with a little hammer, you kick your foot. That's a reflex controlled in the spinal cord. Reflexes are out of awareness. You are unaware of initiating or producing the response, and you cannot inhibit it consciously. We move up. Above the spinal cord, we have the brain. The brain is probably the most important organ in terms of psychological functioning. It regulates thinking, reasoning, emotions, actions, make plans, carry out tasks. The brain has a functional localization feature. In other words, different parts of the brain carry out different functions. Scientists don't fully understand the localization of the brain yet. We only have some inklings about certain functions related with certain brain areas. The brain is a complex structure. To simplify it and better understand it, scientists divide the brain into different areas or into different components. We're going to start from the bottom and move upwards. Right above the spinal cord, right above the neck, the part of brain is called the brainstem. The brainstem is evolutionarily old. When we talk about evolutionarily old, we mean some evolutionary old animals have similar structures and have a similar structure and similar functions. The brainstem, sometimes called the reptilian brain. In other words, we share this structure with reptiles. Reptiles have a brainstems, and reptile brainstems carry out similar functions as a human brainstem. The brainstem control basic biological functions. For example, one part of the brainstem, the medulla, It controls breathing and heartbeat. It also controls blood pressure. Suppose if a person suffers a severe damage to the medulla, to this part of brainstem, what happens to the person? The person dies. Breathing is stopped, heartbeat is stopped, blood pressure is gone, the person is gone. Another area associated with the brainstem, but extends beyond the brainstem, is called the reticular formation. Reticular formation controls arousal and alertness. If somebody suffers permanent severe damage to reticular formation, this person might not die immediately, but he goes into a coma. His arousal and alertness is gone, so he's in a coma. He's unconscious. The brainstem controls very basic biological functions. And another brain area at the lower level, very close to the brainstem, is called a cerebellum. Cerebellum is a Latin word, and it means a small brain. This is a very important component of the whole nervous system. It controls movement. It controls voluntary movement, and it is responsible for kind of making some routine movement automatic. For example, at the beginning of learning how to ride a bicycle, you're going to take a lot of effort. You're going to try to balance yourself on the back. You have to remember how to balance yourself on the back. But gradually, with a lot of practice, riding a bicycle becomes automatic. And you know how to do it. This process for automatization is associated with cerebellum. Similarly, when you are learning to drive a car at the beginning, you're going to exert a lot of conscious control. You are going to purposefully remember all the instructions. When to press the gas pedal, when to hit on the brake, how to turn the wheel, steering wheel, and look for the red light, stop signs. But with a lot of practice, the whole process of driving a car is going to become automatic. It's going to become your second nature. That's how you pass a driving test and get a driver's license. This part of the memory formation, implicit memory or procedural memory, is very much associated with the cerebellum. If we move up a little bit and we go to the middle of the brain, we have a structure called the thalamus. The thalamus is a very important structure in regulating sensation and perception. The thalamus is a relay station for sensory information. Sensory information, most of sensory information first go to the thalamus and then gets diverted in different parts of the brain. For example, visual information go to thalamus, and then it goes to the back of the brain, the occipital lobe. Auditory information also first go to the thalamus, and then it go to the temporal lobe. In the middle of the brain, there's an area called the limbic system. The limbic system, here is the picture of the limbic system. Here you have the brainstem, the cerebellum, and then right in the middle of the brain, you have the limbic system. The limbic system have a few structures that control very important psychological functions. One important structure is called the hypothalamus. Here is the hypothalamus. It's a small structure, not large, but controls very important psychological functions. One function of the hypothalamus is that it controls pleasure. It houses the brain's pleasure center. This was discovered by a Canadian psychologist, Dr. Oates, by accident, almost by accident. Dr. Oates inserted an electric needle in the rat's brain. He found that when he inserted the needle, when he placed the needle in the hypothalamus, the rat likes the feeling. The rat seems to be seeking out the feeling of being stimulated in the hypothalamus. So from this phenomenon, Dr. Oates inferred that the stimulation of hypothalamus must produce intense pleasure. In the rat, how intense the pleasure is, Dr. Oates did an experiment. He inserted the electric needle into the brain of the rat in the hypothalamus, and he let the rat control the stimulation. The rat can press a lever to get electric stimulation in the hypothalamus. Once given this control, the rat would abandon everything else. He would stop eating, stop grooming, not interested in mating. He would keep pressing the bar until he drops from exhaustion. So the hypothalamus produces intense pleasure and produces intense motivation to drive seeking behavior. The hypothalamus has another important function related with motivation. It controls hunger and thirst. The hypothalamus receives information from the blood vessels. The blood vessels contains sensors sensing sugar level. If the blood sugar level is low, the information is recorded in the blood vessels and then sent to the hypothalamus. And then the hypothalamus would trigger a process to produce a sensation of hunger. And this will drive the person to look for food and eat food. The motivation for eating is very much controlled in the hypothalamus. One area in the hypothalamus controls the hunger. It's called the hunger center of the brain. Another part of the hypothalamus controls satiation. When you stimulate the hunger center of the hypothalamus, the animal would seek food, would look for food and eat. But if you stimulate the satiation center in the hypothalamus, the animal would stop eating. It's like a gas pedal and a brake pedal for eating. Hypothalamus also controls thirst and drinking water intake. The information also comes from the blood vessels. The blood vessel monitors the blood volume and blood thickness. If the person is dehydrated, the blood volume shrinks and the blood is getting thick, the information gets sent to the hypothalamus and the person becomes thirsty and seek water. Another really important structure in the limbic system is the amygdala. Amygdala is really important in controlling emotions. This would be amygdala in this area, in the middle of the brain. Amygdala controls anger and fear. It's related with fight or flight. Psychologists and physiologists did experiments to show the function of the amygdala. They opened the skull of a cat, and so they gave they put the cat to sleep, so the cat is unconscious, and then they open the skull of the cat. They use an electric needle to stimulate the amygdala. One part of the amygdala, when stimulated, produce fear reactions. The cat would shiver, the cat would cower, the cat would shrink, and would withdraw. So it seems that the cat is fearful, but the cat is unconscious. This is a pure action of the amygdala. If the scientists stimulate another part of the amygdala, the cat would hiss, the cat would make angry faces, and the cat might even make some attack movements. And amygdala is also related with memory, especially memory of emotionally charged experiences related with fear and anger. Amygdala is very important in understanding anxiety disorders. Why are some people so easily get anxious, get nervous? They might have a very sensitive, very active amygdala. And then this would make them get easily upset, get easily scared, and maybe gets them easily angry. Emotional instability might have something to do with a sensitive amygdala. A lot of research shows, so there are research showing that kids, little kids, 4-year-olds, 5-year-olds, have a lot of individual differences in how sensitive and how reactive the amygdala is. Some introverted kids, some fearful kids, and nervous kids, when they are in a new environment, when they are stimulated with novel stimulus, their amygdala tends to become overactive, tend to overreact. This might be part of the reason they are so easily scared. Still another structure in the limbic system in the middle of the brain is the hippocampus. Hippocampus is very much related with memory. It is particularly related with semantic memory. Semantic memory is a kind of memory we can recall, we can report. It's conscious, it's explicit memory. All this information about psychology you are learning in the course are components of the explicit memory, semantic memory. On the other hand, memory about how to do things, like how to ride a bicycle, how to tie your shoes, these are procedural memories. Most procedural memories are implicit. Remember, procedural memories are more associated with cerebellum. If you do a lesion study, that is, you surgically destroy the hippocampus in an experimental animal, for example, a rat, the rat will suffer memory loss, memory damage. Psychologists have tested this idea using experiments. When you have a healthy rat with a healthy, intact hippocampus, when you make him run a maze, the first time you make him run a maze, it will take a long time for him to find the how to get out. But make him run a few times, he's going to take less and less time to get out. This means that he's learning about the road, the structure of the maze. He remembers things. He keeps information in his memory storage. But if you surgically destroy his hippocampus, he's going to lose the ability to remember the layout, the structure of the maze. The first time you put him in there, it takes a long time for him to get out. You make him run 10 times, he still takes a long time to run out. He has to learn how to get out every time by trial and error. He doesn't remember stuff. This is a water maze. You can use a water maze task to test the rat's or mouse memory. The water is muddy, not transparent, and you throw the rat in there, the rat will be struggling for his life, or a mouse. This is a mouse. And there is a platform under the water. The rat cannot see the platform. The rat, by trial and error, gets to the platform and perch on it to take a break, to take a rest. He is rescued. He finds a safe place. The next time you throw the rat in the water, it's going to take less time. Shorter and shorter time, he learns about the layout of the water maze, and he remembers where the platform is. But if you destroy his hippocampus, he's going to lose his ability to remember where the platform is. You put him in the water 10 times, he still takes a long time to find where the platform is. Hippocampus is very important for memory consolidation. That means you take some information and you consolidate it and move it into long-term memory, so you can recall it later on. This is a picture of the brain. See, you have the thalamus. The thalamus is a relay station for most sensory information. It gathers information from different sense organs and then relay it into different brain areas. You have the amygdala. Amygdala is also in the middle part of the brain. It controls emotions, especially rage, anger, and fear. And then the hippocampus is this structure like a banana. It controls explicit memory or semantic memory. Now we move up. We move up from the middle of the brain to the surface of the brain. The surface of the brain is called the cortex, the cerebral cortex. The word cortex means skin. This is outer layer of the brain. It has all these wrinkles, grooves, ridges. According to evolutionary theory, in the evolutionary history of mammals, the surface area of the cortex gets larger and larger, and the skull remains relatively rigid. As the surface of the cortex gets larger and larger, wrinkles develop to allow more surface area to be fixed in the relatively rigid skull. There are different ways of dividing the cortex. A common way to divide the cortex is dividing it into different lobes. The frontal lobe here on top is a parietal lobe. And then the very back of the brain is occipital lobe, sitting right above the cerebellum. This is back of the brain. This is where your pillow is in the evening when you're sleeping. And then on the two sides, you have two temporal lobes, left temporal lobe and right temporal lobe. These different lobes cover the entire brain area, the cortex area. This is one way to divide the cortex. Another way to divide the cortex, even simpler. You just divide it into left hemisphere and right hemisphere. There's a big groove between the two hemispheres. There's a very interesting phenomena in the brain's control of the body parts. The left half of the body, the left arm and left leg, are controlled by the right hemisphere in the brain. The right arm and right leg are controlled by the left half of the brain, left hemisphere. Suppose if a person suffers a stroke, the stroke is in the right hemisphere and destroy the motor area of the right hemisphere. Which part of the body, which side of the body is going to be paralyzed in this person? The left half of the body is going to be paralyzed. He's not going to be able to move his, control his left arm and left leg. On the other hand, if the stroke destroys the motor area of the left hemisphere, he is going to lose control of his right arm and right leg. This is called a crossover. Now, the two hemispheres are connected by a single band of nerves. This band of nerves is called corpus callosum. If we turn the cortex upside down, we will be able to see the corpus callosum. The corpus callosum is the only connection between the left hemisphere and the right hemisphere. If you cut the corpus callosum, the person will have two separate hemispheres, have a two separate cortex. You might ask, why would anybody do that? Why would anybody surgically cut somebody's corpus callosum? Actually, this has been done in the past. In the past, while in treating people with a severe seizure, which could be life-threatening, neurosurgeons used to cut the corpus callosum to reduce the severity and frequency of life-threatening seizures. So there are patients who have received this kind of surgery. These are called the split brain patients. Their left hemisphere and right hemisphere are split apart with no communications with each other. This graph shows where the corpus callosum is. The corpus callosum is a big band of nerves connecting the left hemisphere and right hemisphere, just below the cortex. The split brain patients who receive the surgery cutting the corpus callosum, they usually survive. And psychologists have observed these patients closely. And they found that in most aspects, they are normal. Their memory is normal, their speech function is normal, but there are subtle deficits. There are subtle deficiencies or subtle impairments in these patients. One impairment is that sometimes the two brain structures, the two hemispheres, give conflicting instructions to their hands and other body parts. The person in the morning is trying to get dressed. One hand is trying to button up his shirt, but the other hand might be trying to unbutton it. So the person fumbles a long time to get dressed. This would be a result of a different command and control, different impulse coming out of the right hemisphere and left hemisphere. There are also some impairments or some interesting phenomena in the way the split brain patients perceive things. Their visual perception is somehow affected. Suppose you show this picture to the split brain patient and you ask the patient to fix it on the red spot right in the middle. You see the information on the left is going to go to the right hemisphere. Information on the left half of the visual field is going to go to the right hemisphere. Information on the right half of the visual field goes to the left hemisphere. You ask the person, what do you say? What do you see? Please name the object or words you see on the screen. The person is going to say, the person can tell you, oh, I see a telephone. He can pronounce, can verbally report, I see a telephone. But he cannot verbally report the word dog. I see the word dog. He sees a word, he sees something there, but he cannot pronounce it, the dog. This is because the information about the word, the visual information goes to the right hemisphere, and the right hemisphere doesn't have a language function. Language production and language comprehension are located in the left hemisphere. In healthy people, in people who have a connections, interconnection between right hemisphere and left hemisphere, information travel freely back and forth. But the split brain patients, they have their corpus callosum cut. So information that end up in the right hemisphere gets stuck there, cannot get transmitted into the left hemisphere, so they don't get pronounced by the left hemisphere. This tells us that language function is housed in the left hemisphere. Another experiment you can do with split brain patients is that you ask them to touch something with their left hand. So, for example, you ask them to touch a pen or pencil with their left hand without looking at it. They cannot articulate what they are touching. They feel something in their hand, they have a sensation about something, the object in their left hand, but they cannot verbally report it. They cannot name it. This is because information, the tactile information from their left hand travel to the right hemisphere and get stuck there, cannot go to the left hemisphere to be pronounced by the language function area in the left hemisphere. But people with healthy brain, people have connections between left and right hemispheres, intact corpus callosum, they don't have this problem. You ask them to touch something with the right hand, the information go to you ask them to touch an object with their left hand, the information goes to their right hemisphere and it immediately travel to the left hemisphere and they can name it. There's some really interesting research done with these split brain patients. You can search them on the internet and it's a lot of fun. Let's look at the cortex more closely. Cortex is very large, comparatively speaking, in the human brain. If we compare the human brain with our closer relatives, the chimpanzee, humans have a much larger brains than chimpanzees. And most of this difference is in the cortex. Humans have larger brains mostly because we have a larger cortex. The cortex controls some really important functions. Let's review a little bit about the different regions of the cortex. The frontal lobe is right behind your eye and your forehead and extends backward to the top of your head. And then behind the frontal lobe, you have the parietal lobe on top of your brain. And then the back of your brain, the pillow area, is the occipital lobe. On the left, you have a left temporal lobe. On the right side, you have a right temporal lobe. We know language function is housed in the left hemisphere. Exactly where in the left hemisphere? This discovery was made by a French doctor by the name of Broca, Dr. Broca. Dr. Broca had some patients he was taking care of. The patients could not speak. The patients could understand what was said to them. They could read, but they could not articulate any words. Their speech function was impaired. After the patients died, Dr. Broca did autopsy on them, and he found that they have a trauma or they have lesions in a particular area in the left hemisphere. Specifically, it's in the left frontal lobe. The frontal lobe on the left side. This area is called Broca's area. This area is responsible for language production, pronouncing spoken words. People with lesions in this area, they cannot speak, and their condition is called expressive aphasia. Remember this word aphasia. Aphasia is a very important word. It means loss of speech. You have to differentiate aphasia from amnesia. Amnesia is loss of memory. Amnesia, aphasia, these are different conditions. The Broca's area. Another important area for speech is the Wernicke's area. This area was discovered by the German scientist Karl Wernicke. Wernicke's area is responsible for language processing. It's responsible for understanding speech language and written language. People with lesions in Wernicke's area, they cannot understand anything said to them. They can still articulate words, but they speak in gibberish. Their speech doesn't make any sense because they don't understand what they are saying either. They don't even understand what's coming out of their own mouths. So they cannot organize their own speech with proper feedback. The Wernicke's area, when Wernicke's area has a problem or lesion, this condition is called Wernicke's aphasia or receptive aphasia. summarize a little bit about the different brain areas we covered so far. The left hemisphere is responsible for verbal processing, speech, comprehension, and reading. And we have to remember, well, two main areas, two important areas are related with speech. The Broca's area is in the frontal lobe on the left side, is responsible for speech production. The Wernicke's area is a little bit behind. It's in the temporal lobe, also on the left-hand side, is responsible for language processing. The right hemisphere are also responsible for some really important functions. There's some evidence showing that the spatial information, spatial processing is largely the responsibility of right hemisphere, like navigating in a new place, finding your way around a new city, spatial processing. This will be in a right hemisphere. And also music perception is mostly in the right hemisphere. Expressing emotions strongly associated with the right hemisphere. More functions of the cortex. When we were talking about neurons, we talk about the sensory neurons and motor neurons. Sensory neurons carry information from different body parts to the brain. To which part of the brain? In the brain, in the frontal lobe, in the very back of the frontal lobe, you have this area called primary motor strip. Primary motor strip. So it will be this strip of the cortex. This area of the brain receives information from different body parts, from the body surface. Oh, no, I have to take it back. I got it wrong. The motor area actually, I have to edit this part. I have to edit this part. And I'm going to give it a space to edit this part. So when we were talking about the neurons, we talk about the sensory neurons and the motor neurons. Sensory neurons carry information from different body parts to the brain. So the brain can process the sensory information and get a sensation, recognize the information. The motor neuron go from the brain to the different body parts which control and command instructions to control the different body parts to make movements, to carry out activities. The primary motor area, the primary motor strip, is located in the frontal lobe. It's near the very end, very back of the frontal lobe, before the big groove that separates the frontal lobe from the parietal lobe. This area of the brain sends out control and command information, instructions to different body parts, the thumb, the fingers, the toes, the tongue, the torso. It controls the movements, motor movements. Of course, the kind of neurons that carry the information to the body parts are motor neurons, mostly. Behind the primary motor cortex, just across the groove, in the parietal lobe, we have this primary somatosensory cortex. We encounter the word soma again. Soma means body. Body sensory means this part of the brain receives information from the body surface in different body areas and it receives the sensory information. receive a sensory information from the fingertips, from the fingers, hand, arms, torsos, all the body surfaces. These are very important brain areas. These two brain areas, the motor area and the somatosensory area, have a very interesting feature. A certain part of the brain is corresponding to certain body areas. If we look at the motor area, we can see a particular motor area controls the toe and foot and legs and trunks. Suppose this is left hemisphere. This is a motor area on the left hemisphere in the frontal lobe. This particular area would be controlling the toes on the person's right foot. And this area will be controlling the person's right foot, right leg, right arm, jaw, tongue, etc. And you can see the part of the motor area that controls the finger and thumbs are pretty big. Finger and thumbs are not that big compared to the torso or the trunk of the body, the legs, but they have a large area in the motor area dedicated to controlling them. This is because the fingers can do a lot of things. You can be very flexible, you can be very clever with your fingers, especially in humans. And then if we look at the somatosensory area, we have the same correspondence between the brain areas and body areas. Each part of the somatosensory area would be receiving information from a particular part of the body. This part of the somatosensory area receives information, sensations from the tongue, the lips, the face, the finger and thumb. You see the larger areas receive information from more sensitive body parts. The lips very sensitive, the tongue very sensitive, the fingers and thumbs are all very sensitive. There's a one-to-one correspondence between brain areas and body parts. Another important function, some people say the most important function, is called the association function, and it's also called executive function. Association and executive functions are mostly housed in the frontal lobe, especially in the prefrontal cortex, that is just below and behind your forehead, above your eyes. This area is basically the command center of the whole person. You make plans, you control your emotions, you carry out your plans, and you self-regulate, you self-discipline, you control your attention. These are very important control and command functions. These are controlled by the frontal lobe. Summarize a little bit about the major functions of the cortex. The frontal lobe, well, it houses the primary motor cortex. Remember the motor strip, it controls movements of different body parts. And then the frontal lobe controls executive function, goal planning, decision-making, reasoning. So you envision different outcomes of different actions and then you compare the pros and cons, and then you choose a particularly good action or your best options to carry out. This is the executive function of the frontal lobe. And you consider the context. Language production is in the right hemisphere, right frontal lobe, that's a Broca's area. Parietal lobe has a primary sensory area. Sense of touch and sensation from different body areas are right here. And there are some other functions. We have not covered those functions in this course. Temporal lobe has a primary auditory cortex. Primary auditory information is processed in the temporal lobe. Comprehension of a spoken language, that's a Wernicke's area. Occipital lobe is in the very back of the brain. The pillow area, this area is a primary visual processing area, processes visual information. There are different ways we can study the brain. One way we can study the brain is brain stimulation. Remember, we covered brain stimulation with animal research. Dr. Oates, a Canadian psychologist, he discovered the pleasure center of mammals, which is in the hypothalamus. He discovered this area by using stimulation. He used an electric needle to stimulate the hypothalamus of a rat, and the rat really likes it. The rat seeks it out. A rat would voluntarily give up food and sex just to get a stimulation in the hypothalamus. And we also, psychologists also use the stimulation method to investigate amygdala. Remember, amygdala is related with fear and aggressiveness and anger and rage. The researchers used the cat to study these functions, and they used the electric needle to stimulate the cat's brain, amygdala, and they can either produce anger or produce fear in the cat, depending where you stimulate the amygdala. Lesion studies also used a lot in animal research. Usually you can't use it in humans. You can't surgically destroy a human subject's brain areas just to do a scientific study. But with animals, you can surgically produce a lesion in certain brain areas and observe what kind of functional impairments appear. If you produce a lesion in hippocampus, the rat usually loses the ability to form long-term memories. Memory consolidation is impaired. The rat can never learn how to, the best way to run the maze. But in rare occasions, lesions are used in humans. In the split brain patients, where brain surgeons, neurosurgeons cut the corpus callosum of people who have a severe life-threatening seizure, this is a lesion study. There is a rationale, there's good reason, at least at that part, at least in those years, there was good medical reason to perform this kind of procedures. It's not mainly for research. Research is a side effect of the medical procedure. And then there are clinical observations. Clinical observation is a very important method. We can observe naturally occurring lesions in humans. For example, those are injured in accidents or disease. And then we can observe what kind of psychological behavior impairments are associated with this kind of lesions. There was one very famous clinical observation completed in the 1800s on, on a road construction worker, Mr. Page, Phineas Page, and this guy was involved in an industrial accident. He got a severe damage to the frontal lobe, and doctors observed him for 7 years. He lived for another 7 years after he got his injury, and the doctors know where his injury is located, the frontal lobe, mostly in the frontal lobe, especially in the prefrontal cortex. And the doctors observed that his memory was intact, his language functions was okay, but his control functions, his behavior control, his executive functions was impaired. He cannot make any plans. He changed his plans all the time. He's very impulsive. He cannot control his impulse. And he cannot show up at his work on time. So he loses ability to make plans, to regulate himself, to carry out purposeful activities. And also he have a difficulty controlling his emotions. He gets angry easily, and he's very abusive verbally. He swears, he curses. Another kind of research, broad types of research, are animal studies. Animal studies, sometimes we use lesion, sometimes we use naturalistic observations, sometimes we use behavior experiments. There are many different ways you can do animal research. We're going to cover some of the animal research, for example, maternal deprivation, research in developmental psychology, later in the course. Okay, now let's take a break.